For those of you just joining, my name is Mike Racht. I'm the co-chair of the State of the Science, along with Dr. Valentino, also the Chief Science Officer at the American Thrombosis and Hemostasis Network, known as ATHEN, and a pediatric hematologist at Oregon Health and Science University in Portland, Oregon. I've been lucky enough to ask to moderate this afternoon's session. With me today is Dr. Peter Marks from the FDA, Dr. Amy Shapiro from Indiana, Dr. Diane Nugent from California, and Dr. Suchitra Acharya from New York. So thank you all for your fascinating talks today. Lots and lots of topics of interest and very all thought-provoking. We had lots of questions come in already. I would request the participants to send any more questions through the website. And I'm going to start with a question for Dr. Marks. So Dr. Marks, in your talk, you gave us an example of how the FDA is easing the regulatory burden for gene therapy. So the question I had for you is, how can this be translated to replacement therapies or non-curative therapies for the ultra-rare coagulation protein deficiencies?
Yeah, thanks very much. So some of the things that are possible with gene therapies or therapies that are based on some backbone of a platform, that is a viral vector carrying them or non-viral vector, allow you manufacturing efficiencies that probably are going to be hard to do in the space of purified proteins. On the other hand, some of the things that we're anticipating using for very rare gene therapies are still applicable to them, which is this concept of clinical trials that are Bayesian designed so that you have every patient contributing to knowledge. So the idea here is that if you have a rare coagulation defect, this is just the same way as you could do it with a gene therapy, you could have every patient have meaning. That is, the first patient that gets treated with the protein-based therapy, before you treat them, you don't know whether it's going to work for sure, right? Your probability of, so in this Bayesian world, afterwards you have a prior probability, then you have a posterior probability, and you can keep working through that. And you do have this advantage with coagulation factors of being able to measure levels. So that also may help here, because you could imagine that there might be the advantage here of seeing us use the accelerated approval pathways as well as efficiencies in clinical trial design. So I think although the manufacturing efficiencies that we might see with the gene therapies might not be applicable here, the other efficiencies such as clinical trial design and use of accelerated approval endpoints might be very applicable.
Thanks. So on a related note, how might this regulatory platform serve as a laboratory for experiments in small clinical trial design and clinical endpoint development? And the reason I ask this is because there are some national health technology assessment bodies that are having difficulty evaluating small trial data for rare diseases and preventing the kind of global uptake of rare therapeutics that would optimize the rationale for development and manufacturing.
Yeah, no, it's a great question here. So I think part of this is starting to make the case here that you can connect the dots, and that, for instance, we're thinking of this like as if we should be able to start to feel comfortable enough, for instance, with our animal models or whatever preclinical models we're using, that when we see something in a clinical trial, for instance, if you see if we understand that a certain factor level is associated with correction of a defect, and we have, and again, for rare coagulation disorders, we don't even need to go to animal models, because unlike some of the places where we're dealing with this in rare disorders, here we usually understand what levels we need. Not always, but sometimes we do. So if we can figure out, what we feel comfortable seeing, hopefully, with kind of a good foundation there, we'll be able to build this so that national regulatory authorities become more comfortable with this concept. It is a challenge because these rare disorders, when with the expensive therapies, they tend to get gun shy often, and that's been a real problem. And so to the extent that we can streamline the development process so that the actual cost at the end of the day is more manageable, that also may help with uptake. That's been the problem, by the way, with rare disease gene therapies in Europe, which is that at the end of the day, European health authorities, and this is not a criticism, they've just decided that the cost, having a 7-digit cost to those is not something that their systems can afford. They can imagine a 5 or possibly 6-digit cost, but 7 digits is just not in the question. So it may be that if we can see our efforts here to streamline development, bring down the cost by maybe an order of magnitude, that may get these to be more acceptable, because then the calculus here, right? The calculus of what the health authority pays to take care of somebody who's managed with a suboptimal therapy or with no therapy at all becomes very balanced by implementing the therapy.
Great, thank you. This next question really is for all of the working group chairs. And as I was reflecting on what I was hearing during your presentation, what I heard was a focus on infrastructure needs for basic and translational and clinical research in the ultra-rare inherited bleeding disorders. So my question for the group is, did you identify specific research priorities across the spectrum that might be used to pilot and propel and build the funding for this infrastructure and research that it needs? And I'll open it up to all three of you.
I can start and have
That'd be great.
the others chime in. I think from our group's perspective, we identified the concept of the use of safety and potential benefit as a regulatory endpoint that could represent a specific research priority across all of the different disorders. As Dr. Marks suggested, what we need to do really is to decrease the cost of bringing anything into development or to market to treat these disorders. So it's not just one specific priority, it's for everyone. In the same vein, harmonizing regulatory agencies across countries and continents can help patients who have these deficiencies worldwide. Prioritizing disorders where there are no therapies at this time or very limited therapeutics could be done. We didn't specifically identify a mechanism to address funding needs to build the infrastructure required. This, I think, will require collaborative work between multiple agencies such as ATHN, NHF, et cetera, to develop the programs that are needed to support these research priorities, including increasing natural history data. And also for Diane's concept from her subgroup about national testing. All of it requires funding to really address these outcomes. And I'll turn it over to Diane and Suchitra.
Thank you, Amy, for summarizing that well. I think for those that know me, I believe in diversity in income streams and support. And I think the only way to find agents and folks that are going to support either a research infrastructure or a diagnostic infrastructure is to say, besides our patients, which is of course why we do this in the first place, besides our patients, who is benefiting from improving the system? So for the diagnostic labs, who benefits most besides our patients in getting to the diagnosis first, to decreasing morbidities, to shortening the time to where the patients get the best therapy? That's all about cost care, health economics. I think that we can create a fund that is almost a pool or a tithing. If we stick with our current system of having multiple healthcare entities, as Norm Faust says, we have 1,500 different insurers. Well, if they would tithe into a pool that helps to support diagnosis, accurate diagnosis, rapid therapy, rapid referral to optimal treatment, insurance companies want that. We just don't have a mechanism for that right now. And I think that we can approach them as a community, as well as the state. Most of our patients, let's face it, are in California, are Medi-Cal, Medicare, Medicaid. Our federal government is also losing massive amounts of money by not getting to the diagnosis right away. So that for diagnostic services, really, really important. On the clinical trials side, I think a lot of funding, if I were a pharmaceutical company, knowing that this work, they're really the questions that Peter and Amy just outlined, health and safety, optimal levels, and maybe it's not a percentage level, maybe it's a clotting activity level, Peter, because we don't, when we're using not factor specific agents, when we're using the other agents, we can't really measure a factor level, right? But what we're looking for is coagulation safety. So who benefits from having a routine, smooth system? Pharma is going to benefit. NIH is going to benefit. We're going to have less money wasted by duplicating services. So to your answer, what I'd really like to do, and I'm willing to fall on the sword for it, is to go out there and get multiple streams of funding, but have them be directed at these different areas. And I think that we will be successful. We'll be able to really drive down the cost of all of this for multiple payers, as well as increase the value and quality of life for our patients.
Yeah, that makes total sense. So along those same lines, Diane, how would you go about establishing these national or designated research centers and the needed biorepository, and putting, without being a cheerleader for Athens, Athens does have the research biorepository already set up. Could that be the beginning of what you're thinking about?
Oh, I think it's already, I mean, in some ways, I feel like that's done. I think we've got that set. There's a place, there's a will, there's financing, the infrastructure is already there. I think more challenging for folks, and Rajiv and I, Dr. Pruthi and I were both lamenting this, is that we both have CAP-CLIA certified labs. We have a research core group, postdocs, that work in our labs. I'm, factor 7 and factor 13, primarily platelet disorders. He has a whole nother panel of things that he's looking at. And if we can get to that, right now, in order to report out a single new diagnosis, CAP-CLIA doesn't really like that much. They're not sure how we should report that into a patient's chart. There's a regulatory panel group that we need to address so that people with rare disorders, if your sample is sent to Mayo Clinic and Rajiv looks at it and he goes, oh, I think let's have one of our postdocs over here work on it. Bam, we nailed it. This is a new variant of factor 5. And you've got to report it. Right now, you kind of have to go around the block saying this is research, this is that. it really slows the whole process down. The data is lost. It goes somewhere in the patient's chart. Nobody knows where it is. And so we need to figure out a better way of doing that. And I think that there are probably 3 or 4 other sites in the country that could be recognized as doing this. This is where you send it for these disorders. You know, that central triage site could really help to direct samples to some of these centralized labs, which again, should be funded so that we can get to this in a more rapid way, a more rapid diagnosis. But there's a problem with the official CAP-CLIA regulated reporting and who's qualified to do that. So that's more of a barrier, Mike, than the repositories. The repositories there. Athens has got fantastic ways to capture the data and the phenotype. We just need to build that up. But I think that there are other barriers that we're going to have to deal with nationally.
Suchitra, do you have any thoughts about this?
Yeah, I think what I would like to see, and I echo what Amy and Diane have already, pointed out, probably establishing a centralized lab at the national level would help, because then what could happen is initially the centralized lab would try to validate all the assays, because currently for the rare bleeding disorders, take PI1 for example, I mean, they're so unreliable in order to make a diagnosis. So at least if you have the central lab kind of do QC, like quality control, and validate some standardized assays, then what can happen is the different centers can approach the centralized lab to get their patients tested. And that way we know we are making an accurate diagnosis. And once you make an accurate diagnosis, obviously it'll help manage these patients appropriately. So, there's a lot still we need to do. And of course, as Diane mentioned, the regulatory stuff is going to be challenging, but with appropriate funding, I think we can do this. Yeah.
So thanks. Let's talk about that funding issue. During your working group discussions, did you have any thoughts about novel funding mechanisms for this infrastructure that you propose? For example, how about allowing 340B earnings to support such activities? I know when I ran our center, we proposed to our institution sharing our 340B funds with some of the underfunded centers in our region, and that was, shot down immediately. Thoughts on that?
HRSA defines the use of 340B as to be used for the diagnosis and access to care for patients with bleeding and clotting disorders. So the cost of diagnosis, which many of our patients cannot bear, their insurance companies refuse to do, that is an appropriate use of 340B funds in order to get patients the right diagnosis. So I think that we have no qualms in doing that. I think more challenging is, I think what we showed in our group is across the country, there are probably regionally, I'd like to see labs get centralized. I do believe that what Suchitra says about having a national center to make sure that the reagents that we're using are all the same, right? So just like in Europe or in England, I forget the name of the institution that is sort of is in control of the optimal reagents and testing and making sure that, a negative falls between this and that this reagent this is what you need to use as control in your lab to do that. We do not have that in this country. It's unbelievable. And so that there's a really important role for centralized standardization of reagents. But I hesitate to have it be one place for all the samples to go to, because what's going to happen there is that the funding that could help regional labs that are specializing, I'm thinking of, for example, the work that's done in Factor 7 by Dr. Shavitz up in Michigan. He has a gene expression lab. He has a focus in Factor 7. that's a good place for a regionalized lab to diagnose factor 7. It wouldn't take much for him to set up the assay and so on. But then when you have a factor 7, he can take it the rest of the way. We aren't going to have a central lab that's going to be able to do that for all of our deficiencies, but we might have 4 or 5 in the country that we could identify, and then that centralized, standardized lab could send to a spoke and hub model for research and clinical trials, not similar to what we do for clinical care.
Yeah. Any other thoughts?
I think 340B funds are definitely something that can get this started and off the ground. I think one of the things you talked about, Mike, from an institutional level is it may not be the center that is the obstacle. It may be the institution parting with the funds or understanding the need to support a national effort. I do think creating an endowment that these entities could live off of rather than year to year or every 2 years or every 3 years worrying about where the funding is probably a more stable concept, but it could definitely get it started.
Right.
Diane, what do you think?
Thumbs up from Diane. You don't have to go off mute to have your thumbs up. Peter, I have another question for you. First, I want to talk about the razor analogy, and then I want to talk about the suit analogy. And when I think about your razor analogy, when I was a kid and would hang out in the bathroom with my dad as he was shaving, right? He had that old-fashioned razor that he would open up the top and drop in a blade and then close the top. Now we have Gillette and Schick and Harry's, and the Gillette head doesn't fit the Schick handle. So how do we avoid something like that happening between companies?
Yeah, so here's actually, this is kind of an interesting, it's an interesting question because it's like kind of a bookend for the one that you asked before. Here is a place where I think things will flip from in terms of gene therapy versus protein-based therapies, because the issue with the razor handle is for right now, gene therapies the manufacturing is so company specific that until something changes, it may be 5 years down the line, it could be 10. Until something changes, to be able to know that when you do a certain setup, you can move from one place to another in the gene therapy manufacturing business. I'm not sure that we're going to be able to let in the gene therapy vector world, the Schick and the Gillette handles get interchanged. However, on the protein-based therapies, if we talk about what we're doing on the downstream end, on the leveraging of clinical trials, such as using Bayesian designs and potentially on using the accelerated approval endpoints and using that paradigm of trying to leverage either an actual level or an activity of a product, there, I think you probably could, that's going to be applicable to something coming out of Schick or Gillette or anyone else, that will fit in on that side. So it's an interesting kind of complementary thing that the clinical endpoints and the regulatory pathway on the downstream end is a little bit more flexible for different protein-based factors than it would be based on our current limited ability to manufacture the gene therapy vectors.
Okay, and then on the, your ready to wear, made to measure, bespoke model. And my daughter just got married and I had my first made to measure suit. Didn't go all the way to bespoke. but made to measure. How do you think something like this would work in the United States, where the profit motive is so important?
So this is actually I'm really glad you asked that question. One of the reasons why we're working with the Foundation for NIH to try to come up with a demonstration project of this is that ultimately, if we're going to actually have this become a viable pathway, we're going to have to show that the efficiencies in this make it a profitable business in some way. And getting back to your recent wedding, I don't know how many people you had at that wedding, but if you went to have wedding invitations printed by an old-fashioned printer, kind of like the printer that my grandfather was, that used to do typesetting and that did high-end printing, it's the setup cost that actually is one of, if you set up to print 25 invitations or 250 invitations or 2,500 invitations on that, on an older, high-end printing press, it's the same setup cost. And so here, the idea here is if you could bring down that setup cost, you might be able to find a way that you could have, they would be the equivalent of being able to make a good living as a boutique printer, printing wedding, small, high quality, small wedding invitations, as opposed to printing like large numbers of magazines that are distributed everywhere. So it is a little bit of the, fine French dining versus McDonald's, to use another analogy, given that on the East Coast here, it's getting towards dinner time. This is really where you'd like to come up with a model that it's one of the reasons why we're hopeful for devices, right? If you could decrease that cost of purification, decrease the uncertainty, so that you could have somebody that's making, I don't know, that would have a portfolio of 20 rare disease gene therapies, knowing that they can't charge like 7 figures for each, but they're making a decent living doing it. I think that's going to become sustainable in the United States. I mean, it's a more of a generics-like model, although generics, I'm a little bit scared to say that, because generics have not been the success that some might have liked them to be. But it is getting more towards that model than the model where, everything has to be, that if you can't get towards 8 or 9 figures profit per year for each product, you're not happy with them.
Thanks, Peter. Any thoughts on what Peter had to say?
Actually, I have a question for Peter. I have $50. I'm going to put it down on one technology versus another. And the part that for someone who sees patients with rare disorders that is so mind-boggling is the diversity of variants. I mean, it's not just one, it's not like sickle cell where it's in one position, it's one amino acid, that's your target. And you can set up the editing. For our patients, let's again take factor 7, it's all across the board. And, in hemophilia, what we've done is to insert the whole gene in a sense that covers a multitude, right, of variants, and just say, okay, we'll hit yours with this one. On the other hand, gene editing, all the way down to the nucleotide, is a different approach where you would kind of dial in the area that you want to edit. It seemed to me that both of those have benefits in the sense that I love your Coke machine or pop soda machine analogy, because in some ways you can see a little guy behind there sort of dialing in, okay, this is the sequence that I want to edit. And then boom, you could just choose it, right? Or alternatively, I want to replace my whole factor 7 gene. If looking ahead that 10 years from now, Peter, where should I put my $50? Where do you think we're headed, and in what group of patients do you think?
I'm hoping we're going to be fit for purpose. And so I would suspect that we would use genome editing where genome editing makes sense, and we would use gene replacement where gene replacement makes sense. What do I mean by that? You know, AAV or similar vectors, you know, they're because they can't, unless you start to make very complicated constructs that use 2 or 3 AAVs, which you can do to get things in, you know, it's easiest when you're dealing with, you know, smaller proteins that you need to replace and deal with that. So it may be that AAV, just don't worry about what the mutations are, just get the clotting factor in, will be appropriate for in some cases. On the other hand, when you have a factor 5, you know, or for that matter, you could even imagine it for, if you know, if for certain other places where you have a very large protein, it's probably easier to just, rather than to deal with trying to get the whole protein in, or figure out whether you can do something like a B-deleted domain factor 8, to just, it may be easier to just go ahead and do genome editing there, because you're gonna have such small families anyway, that you'd have kind of a customized approach there. That might be the way to go. So it'll probably be some mix, and I would hope that's how we'll see it go as we see genome editing come along, in vivo genome editing come along. Probably by 10 years from now, hopefully that'll become a reality. So it'll probably be some mix. And I suspect by 10 years from now, we will have moved on from purely AAV as our workhorse, directly administered vector, to starting to see more, not just other viruses, but also probably non-viral vectors, which may have an increased margin of safety, and also increased applicability, because we won't have this issue of immune responses limiting the efficacy of the vector administered.
Okay, I'll hedge my bet then. I'll go 25 split.
Yeah, that's probably, you know, I think somewhere, I've never been to business school, but I've been around so many people who have been to business school, and the things they taught me are, whenever given that choice, yeah, split the money. And whenever asked the choice of, you know, whenever asked a hard decision, which has a yes, no answer, ostensibly, just say it depends. So those are things I learned about business.
You know what, Peter, I got my MBA, and you just took 3 years, and I could have taken 3 years of study and just like, well, Dr. Marks taught me. So teacher, I got a question for you. I really appreciate your insights concerning the need to develop severity categories and treatment algorithms and guidelines for the use of prophylaxis in some of these ultra-rare bleeding disorders. All of these endeavors are based on the need for centralized data. So how would you think we could incentivize patients and families and centers to enroll individuals with these ultra-rare disorders into the Athens system, so we can use these data for the important needs that you have outlined?
Yeah, so that's actually a very important question. Without data, we don't have access to the number of patients who actually need new therapies that we've just been talking about, right? So I think from the patient perspective, one of the things that could potentially be done is through NHF annual meetings, you know, to incentivize patients and remind them that they need to enroll in Athens if they haven't already. Also, the local chapters could play a role in this, in terms of trying to talk to patients more frequently, since a lot of patients have contact with their local chapters. But again, I think with the ultra-rare group, it obviously becomes a question, how many of these ultra-rare patients do even go to NHF, do attend their local chapters? So there really needs to be an effort from the treatment center perspective to reach out to these ultra rare patients, especially sending out things at our center. We send out newsletters to try to remind these patients if they haven't shown up in like 2 years. We'll say, okay, now you're due for your every other year comp visit, so it's time to really show up. It's only the severes, as we know, do come on a more regular basis to the centers. So that's kind of one aspect of it. From the treater and HTC perspective, I think how do you incentivize the centers is one way to look at it. Is again something which I brought up earlier, maybe a few centralized labs where regionally you can send diagnostic testing to and also try to offer genetic testing. So maybe that may incentivize centers so that they can make accurate diagnoses of these patients. And yet another way may also be to hire coordinators regionally so that those coordinators can help centers enter data into Athens, and that way we can boost data collection for these ultra rare patients.
And just to enlarge on my last question, this is really for the entire working group. Do you think there are any programs that could actually get on the coattails of all the exciting stuff that's happening with hemophilia A and B that could benefit the ultra rare bleeding disorders, instead of creating this separate but maybe parallel pathway for the ultra rare disorders?
I don't mind jumping in. One of the things that we're working on here that could, it's possible that if this goes to leveraging the ability of gene therapies, and with this concept of if you have a manufacturing pathway that's well defined, you can leverage it. So you could imagine that someone might have a factor 9 that they're making. They have a nice methodology made to make that, and they have a backbone vector. If they're able, then if they want to come along, perhaps maybe they want to do factor 7. If they can potentially work in a way that is able to essentially leverage off their initial vector, that may be that may help them start to use this pathway of leveraging off of the same manufacturing pathway, so that for their second, third products that come down the line for rare factor disorders, they're leveraging off of what they've done for factor 9, which is their moneymaker, perhaps, so that the other products don't require the intense preclinical tox, manufacturing, process development, and controls down the line for purification, et cetera. Because what they'd be doing is saying, okay, we did it for our factor 9, but now we're doing everything else. All we're doing is swapping the insert, and everything else we're going to do the same. So that's one thought here. Whether it'll work perfectly, I can't say, but it's certainly worth giving it a try, because otherwise, if we treat, if you have to go back to the beginning for every one of the rare factor deficiencies, it's just not going to be it's just not going to be financially plausible to get there, and we won't get there, at least in my lifetime.
So that's interesting that you say that, Peter, because we actually have seen that already in hemophilia B, right? So Unicure did their phase 1/2 with wild type, and then they wanted to switch for the Padua variant, and they only had to do a 3-person phase 2B trial.
Yeah, I mean, we're willing to so that's the kind of thing because they're basically just swapping out, and those are the kinds of things that the analogy for some of this goes back to what we've been doing for influenza vaccine for years, which is, right, it's almost like a strain change supplement, right? You're not changing anything substantively in terms of the manufacturing process. You're just changing a small piece of the insert, And that means, yes, you have to deal with the potency of the insert, but it means you don't have to start going back and revisiting the toxicology and the manufacturing of the overall viral vector.
Great. Any other thoughts?
Virus.
Amy?
Yeah, I mean, I think one thing we still have to realize is that there are limitations here. Some of these patients have antibodies against the vectors. Not every company has been able to overcome that. AAV9 and many of the others are liver directed. Transducing a child, their liver grows and the effect of the gene therapy may wane over time. And then the level you get may still result in a patient being vulnerable to breakthrough events, in the case of bleeding disorders, bleeding. And what's good about factor 8 and factor 9 deficiency is we have very good therapies to treat those patients if they have breakthrough bleeds. For many of these ultra rare disorders, we may not. So what you're talking about is an end game that's beautiful, but I think we have to make some earlier steps to get these patients adequate therapy before we, as we continue to make progress towards the big end game. Is that a football analogy? Because you know I don't do football.
End game and end zone are almost the same thing. So good job, Amy. Diane, you have a comment?
Yeah, I'd like to see us put under the category of how could we start investigations or research that would benefit more than one single family with one single defect. One of the things we're discovering, which I think is really interesting, there are a few things that are really interesting in when we're doing the genomic studies in patients. But in platelet disorders, and now we're finding in the rare factor deficiencies, that many of the patients are heterozygous. They have one normal gene there. And that there's some reason that and there's a lot of theories about this, imprinting. Why is it that the normal gene doesn't dominate? Why is it this defective gene is the one? And I think that there are areas of research that we could look at that could benefit a whole swath of patients with rare disorders, as well as sort of a different look at how we do gene editing or gene therapy. We know that in cancer therapy, there are certain drugs, 5-azacitidine, most recently with the TET gene and ALL, they're using it to turn on a gene that, was suppressed, and now it's really highly effective. But I think that's a whole nother area of genetic approach that I'm hoping we're putting some funding into. But the more we do, the more impressive it is to me that yes, there are people, especially in consanguinity, that both have really abnormal genes. They're, no protein. That's the minority. The majority of the patients that we're finding are heterozygotes. So what's up with that? And if we could turn on the normal gene in some way, just like P53, all you have to do is have one abnormal P53 and your risk of cancer goes up. Well, where's the normal one going? So we need to just really invest again and overcome the barrier of ignorance surrounding research in this country to start looking at better ways for treating this, because then rare would be a little bit more approachable for what we're finding is about 60% of the patients. And that's true with platelet disorders as well. And so there are ways to answer your question, Mike. What kind of things can we do that could benefit more people? I think that's one of them. I think one great example that's really opened up things for rare is the My Life, Our Future, started in hemophilia and now is trickled down through Athen 10 to rare. It's having a huge benefit, huge benefit. And it's telling us that a lot of our assays are not right. We're finding abnormalities in the factor VII deficiencies that don't correlate with their factor VII assays, but yet they believe, well, that's probably a problem with our assay. We need to start looking at what's up with that, you know? So all of those areas I think are really potentially, what do you call it? High yield, low investment, you know, there's high risk, low, whatever it is.
High
So
feasibility.
anyway.
Yeah.
Yeah, okay, good.
I would like to add to what Diane just said. I think she really brought up an important issue about the heterozygous patients. And we know that in hemophilias, you know, typically levels do correlate with your bleeding symptoms. Whereas in the ultra rare disorders, I think there's a big discrepancy between the residual protein and the way bleeding is manifested, as well as the genotype of that individual. So I think, again, trying to understand what really makes a patient bleed, I think that's probably one of the central questions. And then trying to look at on the genome, on the genetic side, to look at actually things like modifier genes, other genes that may be dictating the clinical phenotype. I think that's something, you know, would really help us. And I think Athens 10 is, you know, at least we're collecting the genotype, phenotype, and at some point probably, you know, look at other modifying factors, which I think really plays a bigger role in the ultra rares as compared to the hemophilias.
So let me ask something to all of you. How do you think that something that we learned from our oncology colleagues and this idea of the basket trials, where at least we're learning safety data from a whole bunch of different diagnoses for a particular agent, could that be generalized to the rare bleeding disorders?
That's a tough one. I think,
Yeah, that would work perhaps for almost for a general hemostatic agent more than, I mean, the problem is you'd have to have something, it almost reminds me of something that would work. Could you find something that's for people with inhibitors that would cross over from different, you know, people with different bleeding disorders that have an inhibitor that would work almost like, you know, something that would be more like a FIBA type situation. I'm just trying. I'm at a little bit at a loss. Maybe
Well,
it's
like
just
the
my narrow mind.
fitusiran is a good example of it.
That's actually what I was thinking about.
anti-AT3.
Fitusiran.
Yeah. Okay. Yep.
And then there's it was just announced a new company was formed called Hemab that is going to be looking at a new bispecific molecule. Initially, it looks like just for Glanzmann's, but from my understanding of the molecule, it could be a general hemostatic also.
Yeah.
Also like the anti-TFPI antibody, I think that's another one which they're looking at hemophilias, which could be potentially applicable to the ultra rares. But I think obviously the safety of these agents is something we don't know about currently. Who knows, you know, how it's going to behave in an ultra rare patient versus a hemophilia patient. So that's, I think, going to be the limiting factor.
I think some of these agents that affect global hemostasis in that way can be difficult in some of these rare disorders in terms of applicability. The level of that agent may not control a particular disorder as well as another. I don't know that one size fits all, you know. So for example, for factor 13, you could increase thrombin generation, but you need a stable clot. For say PI1 deficiency, you need to stop cardiac fibrosis. It's hard. And for some of these disorders, perhaps intermittent therapy for some patients is better than prophylaxis. How do you treat them with some of these agents? It's not so easy.
Yeah, and also some of these ultra rares which have a thrombotic phenotype, like some factor 7s, there are fibrinogen disorders which have a thrombotic phenotype. What if you add some of these general hemostatic agents? Are you going to tip the balance towards thrombosis? So there are a lot of these unanswered questions. So I think we need to wait for the results of the hemophilia trials to get a better idea of what they're doing in the hemophilia patients, just general hemostatic agents, and then see if there's applicability to the ultra rare group. Yeah.
I still remember one of the basic tenets I was taught as a fellow, Diane, probably you too. It's always best to just replace what's missing. Now, there are circumstances like factor 9 deficiency with inhibitors where it's not so simple. And these agents will have a huge impact for those patients. But for some of these other patients, I am concerned.
Well, and this is Peter. I mean, the other issue is that once you go, you see the beauty of filling what's empty and emptying what's filled is that some of the safety issues tend to be quieted down there. What I worry, the problem is with these other things is you start to get into, you're in the very rare space, then you get worried about safety data sets in a very small population gets you more nervous. And there's a little less of that when you're dealing with using the native, we're trying to replace with a native protein or close to a native protein.
So I'm going to ask a little bit more general questions, and this is really for everybody. We hear that one of the barriers that some feel about why they don't go to their HTC is, so many HTCs are at state institutions or they're at government, they're a government-like entity. How do we address this with people who have concerns about sharing their data with government?
They already are. They have a cell phone.
That's right.
That's not a good answer. You have to talk to each person individually and understand their concerns. It takes time. Some people you'll never alleviate that concern.
Yeah, we heard from Amira, the young woman who has factor 13 deficiency at the beginning of this session. She said initially that she and her family were not engaged with the inherited bleeding disorders community. And it wasn't until she became a young adult that she became active with the Hemophilia Foundation of Michigan. Any thoughts on how we might engage this population?
Well, if I understood her correctly, I think one of the issues that she or her other family members may have felt is that it didn't specifically pertain to what they had. Mm-hmm. And so I think a more inclusive organization, which I know Dr. Valentino is working towards, and I know Athen is working towards, will help support that. And again, the treatment centers claiming these rare disorder patients as their medical home, I think is very important.
Yeah. Sukitra or Diane, any thoughts?
The other thought I had is I sometimes wonder when we talk to our patients, what's the literacy level of the patient? At what level do we actually need to explain to them, more like a health literacy kind of approach, where I think it might help these patients actually understand the importance of coming to an HTC. Because a lot of times, when most patients go for their appointments, to their general pediatrics or adult appointments, it's typically a quick, not at the most 10, 15 minutes, whatever, right? But when they come to an HTC, it's a much longer appointment. And at that time, trying to really capture them, trying to understand what their concerns are at their level, at their literacy level. I think we may need to develop some efforts along those lines. And then maybe, that may be something that will help us retain these patients at HTCs and not have them being seen at non-HTC centers. So that's just a thought.
So I want to go back to a topic we talked about earlier in the session when we were talking about leveraging the regulatory pathways for gene therapy for rare diseases. And when we think about what Peter said on how we might be able to accelerate approval of potential therapies for these ultra rare bleeding disorders, one of the things I'd like to hear your thoughts about is, how do you define success in some of these conditions? What would be the primary and secondary aims of such a study where maybe factor level is not what we're really going after?
Who is that directed to?
That's directed to everybody.
Yeah. Well, it's not a new question, right? It came up when Heme Libra first started, started as it's even as a number, not even a named agent. And I can remember sitting at ISTH with people from all over the world saying, well, what are we gonna do? We can't measure a factor 8 for this drug. How are we gonna, know whether it's working or not? And I think we got around that a little bit eventually by, work done by Steve Kitchens and other people to actually be able to measure activities, to look at thrombin generation, platelet, not platelet mapping, but, thrombolastograms and so on. And eventually, I think, people work that we certainly could develop more assays, getting back to what Suchitra and Amy have said, that sort of measure global hemostasis rather than specific. But this has been an age-old problem for the non-factor specific agents, and it's probably going to continue to be. I think another challenge is this issue with gene therapy is for reimbursement. What level should we say is effective? A lot of people would be happy with 10%, but an insurance company doesn't want to pay for 10%. They want to pay for 100% correction. And
Yeah.
so, when they were bringing on the Hep C agent, in Europe, if you weren't 100% cured, they didn't pay for it. So some people are saying, well, if you only get 10% correction, then we're just going to pay 10% for that. So it's not that the level is so uniquely important to the patient or the treater. It's actually going to have an impact on the payer as well. And we need to figure out a new way to say, this is effective. It's effective at 15% correction is what we can measure, but that patient's life is 100% better.
Mm-hmm.
So we can't just keep focusing on the number. We have to think about, another way to measure this. And that's where we have to keep pounding away on getting the patient-reported outcomes of the decreased bleeding, of the phenotypic changes. And I'm worried that if we just keep drilling down on, well, how are we going to measure this one little thing? Kind of like we were all obsessed with 10% or 15% difference in range of motion. And the patient said, that means nothing to me. What matters is I can go out and throw a ball with my kid.
I can grab something off my shelf.
Exactly. And I think we tend to we're very numeric philic. We like to have a number and a correlation, but we're going to have to do better.
Yeah, I totally agree with Diane. I think that fixation on numbers is really not going to get us too far with the ultra rares, because each of these disorders, as we know, patients bleed differently. I mean, if 10% is a level that, say, for example, a factor 7 patients, below 10% would bleed, it's not the same for a factor 11 or factor 13 patient. And so we definitely would need other modalities to look at this, such as viscoelastic testing, as Dan had pointed out, and then to use that to assess efficacy. And of course, patient-related outcomes are definitely important in terms of their quality of life. I mean, how did their quality of life change after gene therapy or whatever other protein replacement that they have received? And I think that really would make a bigger difference than to basically approach payers and make them understand this fact. And that is why I think we need to really define the individual hemostatic levels for each of these ultra rares, and then kind of make a case with the regulatory and payers.
I think, though, to take the other side of the coin, for example, I don't know that you need all of this data up front. I think, for example, when you think about plasminogen deficiency, what we used in the study was basic pharmacokinetics and stability of pharmacokinetics in a very small population to get something approved. Now, once it's approved, we still don't know how to pick out patients who need prophylaxis lifelong versus intermittent therapy. We don't know the optimal trough level for every patient in every circumstance. We will find that out by collecting data over time, say, through the Athens system, and by the patients using the medication. But for some of these patients, having that medication is the first step, which you can do in a very small population, especially if you're looking at potential benefit and safety. Is it causing untoward events? If you've got some good data that shows some basic safety, and that in that disorder there's nothing, then there is potential benefit, and then you collect further data. And I do want to thank the FDA for realizing that in that one very rare disorder.
Mhm. So I know the focus of this group was not on infrastructure, but I think we would be remiss if we don't talk about our workforce and how we can encourage young investigators, young clinicians to come into this field, to enjoy treating patients with these conditions, bringing in these creative young people and having them emphasize this as a career. We might get some solutions that those of us on this panel right now have never even thought about. So let's talk about that. Let's think about how we can encourage our younger folks to move into this field. And that includes you, Peter, right? I mean, how do we help people understand that the kind of training they get through a clinical PT-Monk or adult Hem-Onc fellowship can lead to a really important career at a regulatory agency?
Mike, I don't see that we're having as much trouble in pediatrics as we are in adult. And I don't know, this is probably regional, and different areas have more success than others, but I was at Emory as a visiting professor a few years ago. They're doing great, and they're training wonderful, bright young people. And I know that's true kind of across the country in pediatrics. The challenge that I'm seeing is in adults, that getting a workforce of adults that are being told in their training program there's no money in hematology. You're not going to make it in hematology. You got to go into malignant hematology, I guess is the word that they use. But even at our local university, I was stunned when I went over and said, gee, we'd love to have the adult internal medicine hematology fellows come over and train. And he goes, why would we do that? There's no jobs for them and there's no hope. That was to my face. He told me that. I said, well, I don't know about you. It's unbelievable.
Yeah.
So.
As an adult hematologist oncologist I split my life between clotting and leukemia care. I did leukemia care by necessity at Yale, but the disaster has been that increasingly there are areas of the country that are totally bereft of anyone who really knows adult clotting disorder, a clotting disorders for adults, that pediatricians have to take care of them because there aren't adult hematologists, in part because, look, people vote with their feet. Adult oncology is lucrative, right? So for internists, internal medicine trained adult docs, when they see the lucrative nature of caring for oncology patients and hematologic malignancies patients, there's just not there's not a lot of time left over that for people. They just don't spend the time on non-malignant hematology, and there's because it's just the cost benefit. It takes a lot of time to sort out a lot of the complex clotting factor cases, and so I think they end up just voting with their feet. It really is important, and I think part of the thing that I'm trying to attract people to is, we actually do applied scientific research in our laboratories, so you actually can do non-malignant hematology, but it has to be some combination. I think to just do non-malignant hematology today is a challenge. And so it has to be combined in some way with some type of research often, or some type of other activity, at least for adults. For peds, it's not the same. I agree with you. For peds hemonc, it seems to work very well. But maybe I have a stilted view, but at least that's what I can see. And I can tell you that I still get calls to deal with kind of these emergency in the middle of the night calls from areas of the country where people know who I am. They know I know something about clotting disorders, and they don't have anyone around who knows about a clotting disorder in an adult. And so when they have an 80-year-old who has a PTT of 98 that comes in bleeding, they don't really know how to go about working up an acquired inhibitor. So this is a problem.
Yeah, we've taken care of the adults with acquired inhibitor as well at our local hospital. I think that there's an issue, though, to be blunt, about the difference in what people are paid. And I'm a peds hemonc. I do nothing but hematology. I've been doing it for 30 years. And I signed up an adult hematologist straight out of their fellowship for a salary that I've never made at any time in my career. And so it's one thing, yeah, you're not going to be able to make that salary if you do nothing but heme. But honestly, adult inpatient care for bleeding and clotting disorders, if you generate the team, in fact, there's a lot. I mean, just look at COVID. You think they weren't crying out for a coagulation specialist for adult care? We were the consultants for all the adult intensive care patients. If I'd had a team of adult treaters, we would have been absolutely not having any problem at all in meeting our salaries in the work RVUs. I just think that people need to understand that that's a resource and build it up, just as we have in hematology, that if you build it, they will come. There's a need for coagulation specialists, inpatient, outpatient, women in bleeding, women in reproduction. I mean, it could be totally filled. But personally, I think it's just that we're not offering that or thinking about that for people. I don't think that the business is not there. I think I'm a business person. I mean, I'm in private practice still. And we have 10 hematologists, and they're not doing a drop of oncology. But you have to build destination clinics. You have to show why you can do that. And I think that we've gotten really wimpy at showing how adults can make a business out of it. It's a lot easier just to push some vincristine, for God's sake. So anyway, I'll get off my soapbox. Sorry, Mike.
No, I mean, I think you've hit it right on the head. You know, we have, what, 4 pediatric hematologists on the call. Peter, you're our lone adult hematologist, medical hematologist. And I think the difference between the two is in pediatrics, we all get paid the same. And whether we're doing straight hematology or straight oncology. And in adults, that's very different. Again, reflecting back on when I ran our center in Portland, we had the exact same, I had the exact same experience where we were signing medical providers right out of fellowship who were earning more than me as, 25-year full professor. And, that's not a slam on my salary. I'm very happy with my salary and what I'm earning. But there is a very large financial incentive to not be in classic hematology in the adult world. And when you're coming out with $250,000 of loans and starting a family and having to buy a house, and I mean there's something to that.
But, one of the things that are different between pediatrics and internal medicine is the number of patients that internal medicine hemonc physicians take care of is much larger because people live longer and they accumulate on the adult side.
Yeah.
So the incidence of cancer or malignancies, solid tumors, hematologic malignancies is more than it is in pediatrics. Pediatric hemonc physicians have tended to be based within academic centers because that's where the patients, for the most part, are funneled to because there's fewer of them. On the adult side, the academic centers couldn't possibly take care of all the adult malignancies that are out there in the community. So these people train, they go out in the community, and yes, they have private practices or group practices, and they make far more money than even their mentors who train them within the university. But what Diane said is true. You have to acknowledge that, accept it, and pay people some form of a competitive salary to incentivize them to look at what we do, both on the adult and pediatric side. Another issue that we've addressed is we support a fellowship position. So in exchange for that, we ask that the fellows rotate through our program so that one, at least if they go out in the community, they know something. And two, they know us so they can call us to talk about problems. And every once in a while, you find someone who is interested in what you do and you try to nab them.
Yeah, I totally echo what Dan and Amy have said. It's very, very difficult. I personally, talking about myself, I take a lot of adult, hematologist calls for similar reasons. Somebody's bleeding. There's a mild hemophilia patient who's had dental extraction, now bleeding. What do we do? So that's very common to, a lot of us who treat both children and adults. And I think, ASH, HTRS, ISTH is making efforts in trying to recruit the adult internal medicine folks to go into adult hematology as well as pursue hemostasis and thrombosis, but I still feel there's a huge gap before we can fill this gap. And a lot of also HTCs are, basically managed by pediatric hematologists as the center directors. And at some point, we really need to look at this more closely, because we all have a finite life on this earth, right? And after us, training the next generation, I mean, it's going to be a big issue. And the whole transition process from peds to adults, that's another area where I think we really have to, make some strong efforts to overcome that, so that our patients, are well managed after us. So
let me follow up a question on this training issue. So for people, even people who express an interest in classic hematology during fellowship, do you think that we, the general we, are providing them with enough experience to be able to take care of patients with the ultra-rare disorders, diagnose the patients with the ultra-rare disorders, think about these questions that could be used for advancing scholarly work that would eventually positively impact the lives of people living with these conditions? Or do you think this is something that you obtain after you're done with your formalized training?
I think for ultra rare, none of us leave any training program knowing about that. And that's why I think having this network, not just that the primary care physician can call in, but the hematologist can call in. And, there used to be a site, I think it's still there, called Gene Tests that was offered online. And I would go there almost every clinic and say, where can I send this sample? Call them, say, do you run it for this? I've got this wacky patient. And we really need that. We need that for all of us, to be honest. I know Amy and you, all of us that have a special interest, get calls or emails from people saying, I have a factor 13 patient, they're pregnant, what do I do? And where do I send samples? And what should I follow? And, that call comes to me over and over and over again. And I'm so happy to take it, because at least those people know where to call. And I can refer them to a local HTC. Sometimes I get them from all over the country and say, well, just down the block from you is Suchitra Center. You should go there and, you'll have a local person. We've got to have something that's more organized. I mean, these people just go online and they see I've written a paper on factor 13. And that's how they found me. If we had something that could be globally, just blasted out there so that people would know. And I don't know, maybe a core group of us take those calls once, I'm it for December, or I'm it for January, and all of those calls come in. Or we do, have an on-call volunteer core of us who say, I'll do this, because most of these people end up asking questions, a lot of questions I can't answer. A lot of them I'll say, well, okay, I'll get back. What was that phrase that you said, Peter? It depends. You kind of say, let me get back to you on that, because even I have to find out. But I think that's really important. I do not think we're giving our fellows enough exposure to answer your question specifically, Mike, by a long shot. Not even enough exposure to make them think about it. It's so important to do more training. Sorry,
Diana. Diana. I didn't interrupt. I think I just want to say I totally agree with you that you're not going to have fellows come. I mean, you're not going to have them know about the spectrum of disease. But what I would say from my fellowship training is that it just getting exposure to a couple of cases can light the fire. I remember having when I was a fellow at the Brigham, someone came in. I remember a guy from Maine who had one of the prothrombin, a prothrombin defect that was one of these bleed and clot kind of things, and it was fascinating. And that just gets you, and he actually is pretty funny. It's like, it's really funny to have a patient present that says, oh yeah, and you should look me up because so-and-so reported my, you can see my case report, who sequenced me. And it was, that actually is, that type of exposure is what you need. You're not going to see everything, but I think we need some of that rather than what's happening. I think a lot is in fellowship programs, at least some of the ones that I visited, for adults, it's just boiled down to, they just learn enough to pass the boards, right? They've never seen any of these rare cases. And some of them actually have seen remarkably little hemophilia because they've been so busy in the breast cancer clinic. And yes, they do see, and they go to the heme malignancy clinics and they might see a case or two of hemophilia. So I do think we need to see what we can do there. And again, I think this is an adult issue of trying to find how we can make it possible again for non-malignant hematology to survive. Because I think there is some role for having adult hematologists care for patients in this area, because they do, these issues come up. Lupus anticoagulants, acquired understanding the difference between that and acquired factor inhibitors. Those things, there is something to be said for having people that can still do that, and not have people going, oh, got to call the pediatricians all the time.
Yeah. So Peter, what about, bringing people with this type of expertise into the government or into regulatory agencies? Does the FDA have some sort of program to attract people with this type of expertise?
We do. We try to, and like to get more. We're desperate. We're desperate. And actually, I'll tell somebody, this is like an advertisement. For those who want to do non-malignant hematology, you can actually probably make a living doing it at FDA, because you get the same benefits at FDA as somebody who's heme onc. So it's the whole package deal that you get here. You benefit here because we don't distinguish between non-malignant hematology and malignant hematology. So that's a good thing. And there is this possibility to be able to do applied scientific research. So we have people doing research on coagulation. It's not basic research, but it's looking at impurities in coagulation factor, concentrates, et cetera, how you can make a better recombinant coagulation factor or characterize it. Sorry, Amy, I think you were trying to say something.
I think she was accusing you.
I was saying we need them too. Wait a minute. We're desperate. I'll split them with you.
Yeah.
So as we draw to a close, I think this discussion has been fascinating, and I want to thank everybody. The last question I have, and this is for everybody on the panel right now, is do you have a message for those people who are in the ultra rare bleeding disorders community that we might not have touched on yet, that you want to leave people with, the feeling that I have, this feeling of unbelievable hope, and we're right there. We're really going to be making incredible advancements in this field.
I'd say we care. The hemophilia treatment centers, come in, work with us, help us collect data. It's not always a tomorrow solution, but we can get there together.
Thanks, Amy. Suchitra?
Yeah, I would say if you have a significant bleeding symptom, especially if there's somebody else in the family, which a lot of times is not the case with the ultra rares, it really needs to be addressed. And if you're not happy with your local hematologist or your pediatrician who's trying to work you up, you really need to seek out a hemophilia treatment center. And I think that can actually come from NHF and other organizations that we are involved with, Athen. and basically improve your care. You don't have to bleed, you don't have to suffer, you go through the pain and not and have a good quality of life. So you really need to take care of yourself and partner with the HTCs to improve your care.
Thank you,
And
Peter.
one thing I would say is, at least for our center, is you don't have to give up your local hematologist. We'll work with anyone in the community, pediatricians, internal medicine, local hematologists.
Peter, any thoughts?
Yeah, no, I'd like to try to be, I think I am optimistic that with gene therapies, with leveraging some of the clinical development pathways, our expedited programs, that we can make a difference in people with ultra rare bleeding disorders, because I think it's critical for us to get this right, not just in this space, but there are so many other disorders. So we've got to get this right for so many disorders. And it's part of, I think, what our approach is increasingly, which is a very patient-focused approach in where we're trying to head with what we do at the agency.
Thank you. And Diane, I kept you for last. Diane and I have known each other for 33, 34, 35 years, something like that.
Yeah, it's been a while.
It's been a wonderful
Well,
relationship. What are your thoughts about
thank
this? Because
you.
I can tell you that you have had, everybody on this call has had an incredibly important impact on my career. And your words brought me into this field, and I'd like to hear what you have to say.
You're very kind, Mike. I just, as Peter said, I just pointed the way. You were very enthusiastic. You could have gotten there without me. But
I
I
also
think
had a
that,
full afro back then too. So.
Yeah, right. No, I think what I would want to say to the ultra rare community, which is what your question was, is that number one, although you may feel isolated and like it's just you that has this disorder, come together with the other disorders, come together with all of you, and it's really not that rare. All of us have our own genes, and we really need to come together as a community to advocate, to make sure that there's more services, more access to care, and more support. That's for sure. The second message to the community that I would say, and it harkens back to the speaker, the patient that spoke at the beginning of our ultra rare, and she talked about being in the Factor 13 clinical trial for 10 years. That fibrogammon trial went on for 13 years. And I said to Nord, next time I'm going to choose something in fibrinogen, because that's factor 1. Factor 13 actually linked to 13 years of clinical trial. But yet at the end, there's a product that is changing her life. It's changing people's ability to have children. And so don't hesitate to be part of clinical trials. It really, really makes a difference. And then finally, I'd say don't hesitate to be part of a research question, because a lot of what we're learning now, just from people getting sequenced with genes, or what Suchitra and I were saying before, we're discovering that, well, you can have a disease even though you're heterozygous. You can have a perfectly normal gene on one side and something else is going on. We would know nothing about that except that patients coming together decide that. And I'll just finally say that the first 200 patients that we did with factor 7 deficiency was through a patient-run and led organization. Families coming together themselves, the whole family's coming together to learn more about their disease. This was through CHESS, or the Ladybug organization that has these family retreats. Novo funded them, but the families drove it. So never underestimate your power as a patient to change things. You can do it. And so that would be my message. And to all of us, we need to keep up with the patients and be there for them when they do come together. And thank you, Mike and Len and all of you for your leadership for ultra rare patients, because all of you have really made a huge difference. They call you guys out all the time, as I do right now. Thank you.
Thank you all to all the panelists. I think this was a wonderful discussion. really clarifying for me